Good morning, all. My name is Dennis Lang. I'm the Provincial Pulse and Soybean Specialist for Manitoba Agriculture. Welcome to the Thursday morning agenda. A few housekeeping issues here we're going to start off here with this morning. Please make sure you parked in U-Lot, and if you provide your license plate, in order to prevent your vehicle from getting towed. If your plate has changed or changed vehicles, please inform the registration desk or email immediately. Conference program and presentation PDFs are available on the MAC website if participants want to print them off and follow along. That is important if you're looking at the poster session. You can go on, all the posters are listed online, so you can print those all off as well. For lunch, you'll need your name tag again today for those attending in person. And a couple other things just to finish off here. Big thank you to John Hurd, Marla Rickman, and Vanessa Falk for all the work that they've done in coordinating and organizing the posters. And at the beginning of the coffee break, we're going to have a short pitch from each of those presenters. So let's roll right into it here with our first presenter, Tom Warkentin. Tom's going to be another one of the well-dressed individuals here that we see here today with his VLP shirt. Dr. Tom Warkentin is a professor at the Crop Development Center, CDC, Department of Plant Science, US. Tom is a he's a pea breeder and a soybean breeder as well for cultivars across Western Canada. And together with his graduate students, postdocs, and technicians, he's conducting research to address key challenges and opportunities associated with these crops. The pea varieties arising from the CDC program have greater than 60% market share in Western Canada. Please join me in welcoming Tom to the podium.
Everybody hear me well? Okay. Good. So I want to acknowledge the co-authors of this talk, and I'll mention several of them as we go along. In Saskatchewan, we have broad acres, and this is a picture of one of our field sites near, when I say near, maybe it's like 20 kilometers from the town of Lucky Lake. Have you heard of Lucky Lake? Anybody heard of it in here? Oh yeah, wow, you guys are good. So an hour and 3/4 or so from Saskatoon going south and west. The farmer's land or yard site are the trees that you see in the background, and besides that you don't see many trees. But it's a representative place of the dark brown soil zone of Saskatchewan, and we've been on this farm for many years as one of our off-station sites. So today I want to talk to you about overall goals of what we're doing, and I think part of my theme is to expand our rotations. In Saskatchewan, the rotation is dominated by canola wheat. We'd like to have some more end-fixing crops in that rotation. Here in Manitoba, you're doing a great job sneaking in some soybeans into that rotation, but nevertheless, I think we can still boost up the end-fixing crops in the rotation for better sustainability and so on. So I'll start mainly talking about pea, then soybean, and then wrap it up. So when I look at pea, by the way, this picture is one of my favorites. I took it near Brandon, maybe more than a decade ago. You see more trees, actually. So if you look at sort of broad objectives that I have for breeding pea, at the top of the list, of course, is improving the yield. I think every plant breeder would say that. They want to boost the yield of their crop, and so that's the heavy focus. But in addition to that, and especially when you're applying to federal granting agencies, you need to have a few other nice words in your list. So biological nitrogen fixation, reduced greenhouse gases, and I won't talk much on those, but I will talk about root rot, because root rot, for those of you that are familiar with pea and lentil in the Canadian prairies, root rot is coming top of mind as an important objective. I'll mention protein and heat and drought. I won't talk much about that today, but here we go. So it starts with higher yield. Farmers want to look in the seed guide and pick varieties that have high yield. So I think we have a reasonable track record on that. In this slide at the top, you see the yellow pea varieties, and the bottom are green pea varieties. So on the left-hand side are the varieties that we've released in the last decade or so. And in the two graphs, we have, just get this little thing here. In the graphs, you see the varieties that have at least 1% of the insured acres in the Canadian prairies. CDC Meadow is still the most widely grown yellow pea variety in the Canadian prairies, covering some 20 or so %. It's been at the top for more than a decade. So the turnover of varieties is relatively slow, but it is steady if we can produce something that's good. The other thing you'll see is that all the varieties that you see on this chart are arising from either CDC, that's our program, Crop Development Center, or from Agriculture Canada program. That's led by DJ Bing, my colleague and friend in Lacombe, Alberta. Interestingly, so all those varieties are coming from 2 public sector programs. The trend is that more recent varieties, this is yellow pea, focus on this little chart, more recent varieties tend to be higher yielding. Of course, that makes sense and that should be the case. If it's not the case, then we plant breeders are not doing a good job. So you see here Meadow at about 92% of Amarillo, the Czech in Saskatchewan. But if you look up in this table, you'll see varieties where the yield is 108 or 107 or 109 and such. And then you might ask, well, why do they have less than 1% of the market share? And that's simply because they're recently released. And with pea, a little different than canola, it takes a bit longer to multiply the seed, as you can imagine. Look at the size of a canola seed, look at the size of a pea seed, and there's your answer. So it often takes 3-4 years to ramp up a variety. Of course, this little chart is talking about yield, and for growers of pea, you want to take into account more than just the yield. Don't forget about lodging resistance. You need varieties that stand up well, and that's a big objective in most pea breeder programs. Protein, seed coat breakage, so you have durable seeds that don't crack too much and maintain their germination rate. So within our program, we've released about 40 cultivars since year 2000, and as Dennis mentioned, they're occupying about 60% or so of the area. So this is a snapshot from one of our field trials, our main field trial located just east of Saskatoon. I work with 3 other pulse crop breeders within our group at Crop Development Center. So we're working on 6 crops in total, and several of them are represented here at this trial that we call Sutherland. So we're in a 4-year rotation. This is where our plots were last year, this year, and next year. You may wonder what does that look like for the 2025? We plant onto chem fallow. Why? Farmers don't typically do that. Why? We get droughts in Saskatchewan. Why? That's how it is. So we want to have a little bit of a buffer as plant breeders so that we don't really get wiped out. So we plant onto chem fallow. That's at our home station. When we plant on grower land, which we do a lot, we're typically planting onto cereal stubble. So root rot, you may have heard of it. Aphanomyces is the main culprit here. You get this root pruning, they turn kind of honey brown color. But it's not only Aphanomyces, we have some fusariums as well. So this is what we're trying to tackle. It's complicated. I feel like it's at least as complicated as fusarium head blight for those of you who are looking at cereal diseases. And you may remember that people have been working on fusarium head blight for a long time, making some progress, but it's slow. So yes, have some patience with us plant breeders on tackling root rot in peas. So what we've been trying to do in our program is build on the work of others, and I think that's what researchers should do. That's what we do. We're not starting from scratch here. So colleagues in France and the Pacific Northwest of the US have had this root rot, especially Aphanomyces problem, for 20, 30 years. And so we're kind of building on what they've done. So I'm going to talk a little bit of breeding genomic kind of language here. I hope that's okay, and I'll try to summarize things. But basically, our colleagues have found these different QTL. So QTL is quantitative trait locus. So it's sort of a region of the genome, a chunk of chromosome that's associated with something. In this case, associated with Aphanomyces resistance. And there's at least 7 such locations in the pea genome. So it's not simple. It's not just one great spot. And these 2 in orange, they call them major QTLs. they have the bigger effect. So we thought, let's try to move those major QTLs into material that's adapted to Western Canada. So we have a crossing program to introgress the so-called 7.6 QTL and a separate one to introgress the 4.5. And then once those two, then we merged those two, we crossed those two and carried it forward. So the idea is to kind of pyramid is the word we use in plant breeding language. Pyramid favorable QTLs in this case for resistance. So what has that done? All this work we've done in collaboration with Sabina Benitza, who's in our group, plant pathologist working especially on pulses. So if we start with meadow, the most widely grown variety, it's showing like 80%, 85% root discoloration in this indoor assay. We cross, and so that's our standard. Here, these varieties in red are the CDC varieties into which we're trying to bring the resistance from these lines in blue. And coming out of that, we have some of these lines in green. These are a selection of those. And you see that there's a progression toward lower amount of root discoloration. You also see it never hits 0, it doesn't even hit 10. But at least 30 is better than 85, right? So that's the direction we're trying to move. So what have we done? The best of these we've put into yield trials in the field. We start to make breeder seed from those. We even put them into the so-called co-op registration trials. Tammy knows what those are. These are sort of the official registration trials that breeders use prior to asking for support for registration. Having said all that, well, let me show you a little bit about a root rot nursery. Here's a aerial view of a root rot nursery that we established at Saskatoon. It's 1 acre. We use about ½ acre each year and then rotate, so a 2-year rotation. We make the plots as small as possible because we want to jam in as many as we can. We're using this for pea and lentil. It's labor intensive because we actually dig up the plants and wash the roots and score them for disease. And it took a while to develop a good nursery, but it is quite good now and we get plenty of disease. So in addition to our nursery at Saskatoon, we're working with colleagues at Morden and Lethbridge, also having root rot nurseries. And so what we did here on this top panel, we put the 20 or so lines that we have, that our program has in the so-called co-op test. We put them into these 3 nurseries, see what happens. And sure enough, there's lots of disease, you know. So we've got the disease levels from Saskatoon, Morden, and Lethbridge, and then the average of those 3 numbers is the little black diamond here. So over on the far left-hand side, some of these lines with the really long names, those are the ones that are arising out of our molecular breeding approach. So we feel like, okay, we've made some progress. We have less disease here than we do with these varieties here. So that should be a success. But to cut to the chase, neither of these 2 lines had enough yield in the actual co-op trials, which are conducted under perhaps less disease level conditions. So I don't think we're going to go ahead with either of these first 2 in this first cohort. But we have better stuff coming. We've made many more crosses. This first set was kind of a pilot project, so stay tuned. And then you could say, in my mind, a little bit as a stopgap until we breeders bring you something that we're really proud of in terms of disease resistance. We decided to test all the varieties that are in the seed guide at these 3 nurseries. And just with the thought that maybe some are a little better than others, and farmers could use some of the varieties at this end of the scale rather than the ones at this end of the scale in the intervening years. So we conducted that in 2024. We're going to repeat it in 2025. We'll summarize all the data, and it'll be used to add to the information in the provincial seed guides, so that growers have a better indication of which lines they might want to grow, which varieties they might want to grow, if they feel that root rot is a big issue on their farm. So switch gears a little bit. I want to tell you about some research work associated with pea that we're using to try to address root rot, protein, and other traits. So we developed this panel that we call GWAS. It stands for Genome-Wide Association Study, and it consists of 255 accessions. Accessions could be from gene banks, it could be varieties. It's very diverse, as you can see here. It's coming from commercial breeding programs around the world, it's coming from gene banks, and it includes commercial varieties from our program in Ag Canada. So what we're doing, we thought, well, okay, let's evaluate this panel and see if we can find more resistance to Aphanomyces. So Davini here, our colleague, evaluated them indoors multiple times, multiple reps, and here's what we found. Out of the 233 lines, I mean, the majority of them, or the bigger set, fell within the 60 to 70% disease level, but you can see there's a few that had lower disease. And then when I looked at just a few of those 233, some of the named varieties, here they are. So you see Meadow, yes indeed, it's still in this 80 plus category, but you see some over here that actually have a bit lower disease. And that made me think, when I saw this result, I thought we should bring that to the field and can we confirm that result and show that certainly some of the varieties are actually a bit better. We want to go here into the future, but anyway. Here's another graph, and I'm going to show you 2 or 3 of these, and don't worry about too many of the details at the bottom, but just look at the beautiful colored picture here. This is a genomics kind of a diagram that my friend, colleague Kishore put together. And so basically, we're associating the DNA profile of these lines with their response, in this case, to Aphanomyces. And when we see little dots up at the top, those make us excited because these little dots represent a single spot on, in this case, chromosome 2, that has a high likelihood of being associated with some amount of disease resistance. So then we can create a DNA marker associated with this spot and help us use that marker when we're making crosses and selecting the progeny from those crosses. And this is a logarithmic scale, so something up in the stratosphere here is actually something that's quite important. We also screened our material for Fusarium avinaceum. I told you that Aphanomyces is not the only culprit, so Fusarium avinaceum is also important, and that Fusarium complex. So this work is being done by Davini, and here's Stephen. Stephen Awadelli is a PhD student working with me, and his focus is completely on Fusarium avinaceum. So the story here, when you look at these 233 lines, a bit different than Aphanomyces. You actually see quite a few that have quite low disease levels. But, there's always a but in research, and that is basically all of these here with the high disease resistance are purple flower types with a pigmented seed coat. So we see a big association with this pigmentation and resistance. But in pea production, almost all the pea production is white flower types with light seed coats. So can we find something kind of in this range that has white flowers and at least some moderate resistance? And that's what we're hoping to find. Likewise, when Kishore does this GWAS analysis, again, we find some significant markers here related to Fusarium avinaceum resistance, but we think likely these best markers are probably pigment-related markers. So we need to drill into that further. This one is, I'm switching gears on this one slide related to protein, and when we were driving here today, we talked about Roquette, and they're isolating protein from peas to go into the food industry. Hopefully they're paying premiums to farmers who grow high protein peas. So there's some buzz, some excitement about plant-based proteins, about pea, especially in that market, faba bean also. So we've put a fairly big effort on looking at pea, at seed protein concentration in pea. And when we studied this panel, we found 3 markers here that really stood out quite well in terms of being associated with higher protein concentration in pea. And in the bottom here, Kishore has made these 3 little graphs. And so basically in each graph, it's showing that if you have this allele instead of this one, you get, about 1 or 2% more protein. It doesn't look like much on these graphs because he's got this on a 0 to 30 percent protein scale, but adding a percent is worth a lot, I feel, in that. And if we can add all three of them, maybe we add 2 or 3% to the total. So that's a topic area that we're working on. Again, looking out a little bit in terms of research work, the next 3 slides, I think I'm going to show you about 3 different research-related activities associated with breeding, but really on the research side, developing populations that we hope will push us towards these goals of higher protein and yield. So in this case, we call it NAM, Nested Association Mapping. It's a situation where we cross one variety that we like with several others and we make separate populations. So we have 15 separate populations, a total of 780 lines. They're now already at F8, if that rings a bell with you, 8 generations after the cross, so that means they're very true breeding, and we're evaluating them now in the field. All of these we've genotyped, so we know their DNA profile. We combine that with their protein and yield profile, and hopefully find either directly lines that are good as varieties, or at least good as parents, or at least good markers that we can use to make future varieties. Another approach we call MAGIC, and I forget the exact acronym, but it's a multi-parent panel. Again, thanks to Kishore for doing this. You can see I acknowledge him a lot. He's a fantastic molecular breeder. And actually, we developed these populations. Basically, we crossed 8 varieties of our interest. 2 of them are high-yielding released varieties. 3 are high-protein kind of breeding lines, and 3 are lines that have good root rot resistance, but they're not really adapted. They're coming from the gene bank. So we cross them in all possible combinations. So you have 96 combinations. And then we've sequenced them at a low depth, and they're going up. They were out in the field already this summer. Again, the goal is higher protein concentration and better root rot resistance. And so actually we made this in Speculate on spec a couple of years ago, and then a Genome Canada project came along with funding from multiple partners, and so we fit into that. And the last one I'll just mention from this sort of theme is that, when all else fails, we create some mutants. We used a mutagenesis approach if we can't find the diversity within known pea varieties or germplasms, maybe we can create some new diversity. So we worked with the University of California, Davis to do this fast neutron bombardment of seeds of a variety called Tollefson, which is a recent variety from our program. And we're going to bring that along and genotype it, phenotype it, and hopefully find something interesting. This is what some of those plots looked like this past summer. Again, just east of Saskatoon. In this case, these are what we call microplots. They're 3 short rows, 1 meter long, and that way you can fit a lot into a given area. So even this MAGIC population, there's 2,700 little plots in this area. And you might think, what's this pattern with this darker green blobs? It kind of stands out. So I mean, who wants to take a guess what those little blobs are? Not kochia. Did you say kochia? They're soybean actually. So these are all pea plots, but we put soybean as a marker every 20th, just kind of so we know where we are in the field. But it stands out quite nicely in this kind of an image. Going a little bit exotic, plant breeders like to, okay, I gotta get going. Here's a winter nursery. We're growing peas in New Zealand. I haven't even talked about soybeans, so I'm gonna go quickly. Here's Katema. Katema's working with me as a soybean breeder, and we're collaborating with a bunch of people in Canada and US. We're at the frontier for soybean. We want yield, we want maturity group 00 or 000. That's our key. Light Hylam color, so it could go into food markets. Amazingly, we can grow soybean in Saskatoon, which is similar latitude as Swan River, those of you here in Manitoba. So soybean will do well with water in August, right? August rains. We'd love them, but irrigation would help. We thought at first that maturity was our number one issue. I feel like now we've kind of got that under control. Now we want yield to go with that earliness. We can even grow soybean at Rostern, another hour north of us, so imagine an hour north of Swan River. We released our first variety, we call it CDC Cedar. These are data from the Saskatchewan regional trials. It's not bad. I'd say it's reasonable for yield and maturity, 2 days earlier than OAC Prudence, kind of similar to AAC Halley. And Dennis kindly put it into the Western Manitoba trials, and I think it did decent there as well. -4 in days to maturity, kind of similar to Halley and Siberia. So we're moving in that kind of a direction. It's a relatively small program so far, relatively young, but I think we're making some progress. Here's the breeder seed production of CDC Cedar at New Liskeard in Ontario. It's going to be represented by Secan, and this photo was taken in mid-September. Again, a little bit exotic when it's -30 out. I think I like looking at things that are green, and so we've sent some soybean F2 populations to Costa Rica. You can see at the end of November they already had nice pods. They're going to grow 2 generations for us in the winter. I feel like that's kind of impressive. And last kind of data slide, and I'll get a little bit techie on this one. We're trying to use genomic prediction to help us simple-minded plant breeders make the right combinations. Because you imagine you can cross this with this or this with this, and there's so many potential combinations. So we did a simulation with about 5,000 combinations. These are the best 1,000 of those. And so what we're going to do is we're going to pick these that have high yield and early maturity. Let's call that 000. And high yield and early, a little bit less early, medium maturity, call it 00. That's what we're trying to do. Will it work? I don't know. We're going to give it a shot. We've been moving down this direction since 2022, so stay tuned. So I'll wrap up with that, thanking many of the technicians. These are, let's say, our senior technicians in our group. These people have all been in our program for 10, 20, 30 plus years. Also many other colleagues that I work with in Saskatoon and broadly, and funding organizations. And not to overlook Manitoba Pulse and soybean growers, thank you. I'll wrap up with that.
So we do have time for any questions from the audience. If I think Ann Kirk has a mic here, so if you want to raise your hand, Ann can run out to you quickly and ask questions. One from Dr. Bullock at the back here, so we'll just get him up there. And maybe while we're doing that, I'll get Marla to read out the one question online as well.
Sure. Which soybean varieties in Manitoba have low rates of cadmium uptake?
Low rates of cadmium uptake. Please contact Dennis Lang. He's standing beside me.
That's a question for after the conference. So, and oh yeah, go ahead.
Like my guess is that cadmium uptake is low for all of them.
Yeah, I guess there is one area that they are looking at higher cadmium uptake more kind of west of the escarpment, but that's further research needs to be done on that yet. So, and Dr. Bullock.
Hi, thanks for the presentation. I'm going to go back to a slide that caught my eye. We were looking at the root rot at the 3 locations.
Yeah.
And Family's Farm is north and west of Saskatoon, and I noticed that Saskatoon was always high compared to the other, or almost always high compared to, what were the other ones? Lethbridge
and Morden.
Morden. Why is that, or do you have any idea why that is?
Yeah. So that could be a little bit artificial. All 3 nurseries have good disease pressure, but part of it is just the timing of when the data were collected. Because waiting 1 week later, the disease goes up. Going in there 1 week earlier, it's less. It's really a timing thing. So, yeah. If we went back 5 or 7 years ago, the numbers for Saskatoon would have been quite low because we started a nursery from scratch about 7 years ago. But we just went continuous peas in that nursery, and amazingly, with some water, the disease built up.
Okay. Thanks, Tom. Yeah, so we'll just join me one more time in thanking Tom for his presentation.
And I forgot to thank Dennis for the wonderful shirt. I think I got the nicest shirt in this room.
And Jared already has a photo, so just so you know.